Lecture 6 in the series is focused on a brief survey of reconstruction of metabolism in eukaryotes. So we're going to talk about a few classes of applications here, not detail any one of them in particular, but do a little bit of a survey here. So Saccharomyces cerevisiae, as you know, is a very popular organism. It's been studied extensively and was kind of the lead eukaryotic organism in this field. We do now have reconstructions of unicellular eukaryotic algae. This will be Chlamydomonas reinhardti, which is a model organism. We now have, as mentioned in an earlier lecture, a global reconstruction of the human metabolic network, which accounts for all the metabolic capabilities encoded on the human genome. And I think, as I mentioned before, all of these capabilities are not used in every tissue, so you normally have to customize those, the global reconstruction based on expression profiling, proteomic data, and so forth, that is specific to a cell type or a tissue when you build a network for those cells and those tissues. So you can build multi-cell models like that, that live in the microenvironment in tissues and tie them together. And this is not too dissimilar from trying to study microbial communities like I talked about in the previous lecture. You can also build more coarse-grained models of a whole organ and then link them together by exchange with plasma, and we'll show that. I would say that this last bullet point represents kind of early stage efforts, but some successes have been achieved with this application. So if you like cheese, bread, or wine, or all at the same time, you will like Saccharomyces cerevisiae. So Saccharomyces cerevisiae and its metabolism has been reconstructed through a succession of reconstructions that's illustrated here. Little different history than with E. coli because it bifurcated into multiple parallel efforts, as the series of blue boxes shows. So there's a timeline going down the middle of that diagram. So the yellow boxes represent pre-genome era reconstructions of Saccharomyces metabolism. And they are in yellow, and a few of them were done in the mid-90s. Then after the genome came out, the sequence came out, there was a model built and published in 2003, as I recall, called IFF708. So FF stands for family and Forster in that case. The last names of the two people that built this together was a European, US kind of collaboration, if you will. And that set in motion a process of building multiple different models of it that have now kind of converged to Yeast 4.0, at least published as of 2012. I think there is Yeast 5.0 now and Yeast 6.0. And I think that the community is getting close to having a consensus model of yeast metabolism, although some disagreements still exist. Down below is a diagram that shows the gene count, the reaction count, and the metabolite count in these reconstructions. And that diagram is very interesting because you see it's very low for the pre-genome era models, and then it jumps up but stays relatively flat after that. The green line is the gene count. So in the first one it was 708 genes, and in the last one shown on this slide is under 1,000. It's like 980. And I was always puzzled by this. Why are there more metabolic genes being found in yeast and placed in this reconstruction? So yeast has 6,000 genes or so in it, E. coli maybe 42 to 4,400. But E. coli metabolism is much more diverse. And if you remember that reconstruction, the gene count kept growing and growing and growing and growing. And it's now at close to 1,400. This has not broken 1,000 yet. And I was discussing this with Steve Oliver from Cambridge, and he said you shouldn't expect it to go much higher because yeast is actually a fairly specialized organism in its metabolism. It lives in the leaves of grapes, and is ready to convert sugars into ethanol. It has a much more restricted range of metabolic capabilities than does E. coli. And I believe also its temperature tolerance is also more restricted because it doesn't often go to body temperature to 37 degrees. It's more specialized to ambient temperature, maybe in the 20 degrees centigrade range. So this is a pretty deep history here of these models, and the yeast model has been used to study a lot of different aspects of yeast physiology. Been used to study gene functions, been used for metabolic engineering and alike. I thought I should mention this following this slide, this notion of jamborees, following the previous slide. With the E. coli model, it was done successively by basically an individual driving every reconstruction, every expansion, and curating it. The yeast history is a little different. It is characterized more by group efforts, and in the late 2000s, this notion of a reconstruction jamboree was formulated, like a sequence annotation jamboree, where you get the experts together into a room for a couple of days or so to help curate the literature and have every domain expert come in and share their knowledge, and then formalizing it through a reconstructions with all the reconstruction process with all the QC, QA steps that are involved. So a reconstruction jamboree was carried out for yeast, for human, for salmonella, and I think TB, and some others. Maybe there are now some online methods of doing this, which are maybe easier to implement. But I think still every once in a while the community that is interested in a particular target organism needs to come together and have face-to-face meetings and discussion, debates, delineation of maybe the most difficult issues associated with the curation. The ones that are automatable or easily retrievable from the literature and easily reconciled, maybe they can be done in some community fashion, through the internet. But I think face-to-face meetings ultimately are also required. So the Recon 1, the first version of the human metabolic map, was done by 7 people here at the UCSD campus. Recon 2 was done in a jamboree setting, and I think there are between 30 and 40 authors on that paper as an example. Okay, so let's first say a few things about green algae. Okay, that went by a little fast. But Chlamydomonas reinhardti is a unicellular algae. What did I say? Chlamydomonas reinhardti is a unicellular green algae that is widely studied, including here on campus. Some of you know Steve Mayfield, that has studied this organism for a long time and has built industrial biotech around it. There are other good model organisms for green algae, particularly Chlorella vulgaris, which is a very well-known organism, and its sequence is now available. So when you build reconstructions of a eukaryote, you have to compartmentalize. And in green algae, there are many compartments. I forget now how many compartments are accounted for in the yeast reconstruction. I think it's something like 6 of them. And so you see a number of compartments in green algae. Of course, they have mitochondria and the chloroplast, so they will have more compartments than a yeast would. One interesting thing about this reconstruction was that the photosynthetic apparatus was detailed painstakingly, like the Synechocystis reconstruction. And since the absorption spectra of many of the molecules involved in that is known, you could actually make predictions on how the photosynthetic system responds to a photon as a function of its wavelength. So chlorophyll mostly absorbs at 660 nanometers, but light exists outside of that range, of course, and is maybe captured with different efficiencies, and you can now study that process in this reconstruction. And this is another one of these membrane processes that has been detailed. So a model like this has some capabilities to predict growth rates as a function of light intensity, for instance. So light and its quality is a key substrate for photosynthetic organisms. And what is shown in the bar charts there to the right is the yield per photon from different light sources. And you see these tall bars furthest to the right. These are growth experiments done with LEDs, which have a pretty narrow emission spectra. So you can get an LED that may emit at 680. It's actually 680 where chlorophyll absorbs. And then they may have a 20 nanometer window or so, so you can pump light in at that wavelength, where you get very high efficiency of absorption. And also in terms of the conversion of electricity into light, LEDs are incredibly efficient in converting electrical energy into light energy. So therefore, people have been building LED-based photobioreactors to get maximum efficiency of this process. It's expensive instrumentation, so if you wanted to use that, scale it up or whatever, it would have to be for a high-value application. It is interesting that when you start building those reactors, you find out that the 660 nanometer LEDs are the cheapest. 680 is very expensive. That's because the sensitivity of the human eye starts dropping off at 660. So when you buy, you see the LEDs in your electronic equipment, they're emitting at 660, where you can detect the signal well with the human eye. So people have evolved photosynthetic green algae to grow at 660, and they reasonably quickly adjust to that. They can use that quality light. So reconstructions can help you with these kind of issues. So let's say a little bit about humans. I've already mentioned Recon 1 several times. And this image was actually shown earlier. So this is the process that went into building Recon 1 with the 4 steps. And the validation step, in this case, the last one, addressed 288 functional tests of human metabolism. And through this iterative process I mentioned, they could all be reconciled, and Recon 1 can recapitulate those 288 metabolic functions. So Recon 1, so let me say a little bit about the name. Why is this called Recon 1? So when the human genome was built, it was done in iterations. So there was build 1 of the human genome, there was build 2, and so forth. And when it got to build 35, we had 99.99 some percent coverage of the euchromatin in the human genome, where the genes are. And that's where that was a point in time when it was decided to do the reconstruction. So I don't know what build number is currently on the human genome, but presumably it's higher than 35 now. So since this was done by multiple people, the naming convention we had before, IXX and a number, didn't work because it was done by many people. So it was decided to follow that build X convention and build Recon 1. We now have Recon 2, and hopefully a process will soon be put together to build Recon 3. So it's a multi-scale knowledge base, as we talked about before. Some of its features shown here. There's the gene reaction, gene-protein reaction associations. On genome scale, there are the reactions and the metabolites, the chemical properties, the proper stoichiometry. Map is available. It's pretty large. Just under 1,500 genes accounted for in here. And the compartments it has is shown at the bottom. There is 7. And this table shows a little bit more about the compartmentalization, the reaction metabolites that are in the different compartments. And as I mentioned, the same metabolite may be in many different compartments. So when you look at the stoichiometric matrix for Recon 1, malate, for instance, there will be multiple rows for malate, but one is in cytoplasm and one is in the mitochondria and so forth. So therefore, people talk about unique metabolites in these reconstructions for multi-compartmental organisms, but then there's the total, and they may be bigger. But that's because the metabolite may appear in different compartments and actually have slightly different functions, so they have to be separately accounted for. And I'd like to emphasize that's another measure or notion of location in biology that's very important. The other that I've mentioned a few times is genomic location, where on a genome a gene is. It's very little to do with the XYZ coordinates that we are so familiar with in physics. And you also see here that the protein content of membranes is explicitly represented, and some of the reactions are associated with a membrane. No metabolites in a membrane, but the reactions can take place in a membrane, and they may act on metabolites on either side of the membrane depending on where the reactants or the products are located. So this reconstruction is very large, very detailed, very careful, very extensively supported by the bibliome, and it's not been used in some of the previous slides where we are comparing reconstructions because it's in a category of its own. So this slide I also showed. This is the category of uses. Briefly, the tissue customization that I mentioned, the mapping onto other mammals that is not as successful as one would have hoped, and then the analysis of different perturbation of metabolic states, particularly by either inborn errors in metabolism, where a Vmax through a reaction is severely affected in an individual that may have a detrimental mutation, or a drug intervention, or a toxin intervention or something like that. And you can actually have cells side by side, treat them and not treat them, say, with a drug, and then compare the responses. Or you could have a normal person and a person with a genetic lesion and compare the responses. I should mention that there are many different algorithms now available, and we may talk about them later, to do this tissue specialization. And typically this algorithm takes Recon 1, then takes the expression profiling data or the proteomic data and maps it on there and customizes it. For a cell like the red blood cell, this is very easy, or the platelet, there's no nucleus, so there is no mRNA. You do a deep proteomic profile, then you can map those peptide fragments onto Recon 1 and build a functional model. And that's also enabled in part by the fact that the metabolism in those cells is relatively simple, it's highly customized. So that's an example of specialization. If you get into doing this, I want to draw your attention to that paper at the bottom of this slide. This is a paper that looked at all these methods and applied them to the same organism and the same data sets and did a comparison of their pros and cons. There was a little bit of, my method is better than yours in the literature. So this third party kind of came in and looked at them all and tried to do an objective assessment of what the pros and cons are. So here is a study that Nathan Lewis did some years ago, published in Nature Biotech, which is to get at this issue of the tissue microenvironment. So all human tissues are comprised of functional subunits, like the nephron in the kidney that is well known. And and then the kidney, I forget now how many million, 6 million nephrons or so in the human kidney, as I recall. But the nephrons are the same. They are comprised of several different cell types that carry out different functions. Same thing in the brain. There are the neurons and different types of neurons. Then there are the astrocytes and glia and so forth. And there is metabolic subtasking amongst these cell types. The astrocytes, for instance, help support the neurons in their function because the neurons are very specialized in transmission of potential. So it is of great interest to be able to expression profile with high degree of specificity the tissue types in a microenvironment, reconstruct their cell-specific metabolic networks, and then interface them to get at this metabolic subtasking in the microenvironment. And this is one of the earliest, or maybe the earliest, study that did that. And the steps in here are shown pictorially. First, you have to do the global reconstruction, which is Recon 1. Then you have to customize it for a particular tissue. And there we see an image of a microarray that indicates that there is expression profiling data being used to customize it. And then you build a model where the export from one cell is an import into another. And you have an interstitial fluid in between, and you build a model with these compartments. And then it's ready for use, and as shown at the bottom of this slide and detailed in this paper, this was good for gene discovery and implicated a gene involved in Alzheimer's, because in Alzheimer's, one of the classes of neurons selectively is depleted, and it has a different metabolic coupling to the supporting astrocytes than the type of neurons that are not depleted. So that was kind of interesting. You can analyze expression profiling data, maybe with age and so forth, to see how these functionalities change. And you can also then correlate the outcome of these computations with overall observed physiological parameters. So that's the type of models that really need to be built for the major human tissues. There is an effort to try to build cell-specific models for all the cell types in the human body. And Jens Nilsson at Chalmers in Sweden, Chalmers University in Sweden, is trying to do that. There are other groups trying to do it. Ines Thiele, now at the University of Luxembourg, is trying to do the same. So there's a lot of effort in trying to do this. Oddly enough, not a lot of funding for this activity yet. You can make also a more coarse-grained model of systemic metabolism in humans and do it more at the organ level than at the cell type level in the tissue microenvironment. And here is an early attempt in this direction to interface the liver with skeletal muscle and with the adipocytes, fat. So these three things interact in the body quite extensively metabolically. And that table that has all these detailed numbers just talks about the specifics of the reconstructions, but the bar chart there at the bottom is more interesting. It color codes the composition of these cell types. So yellow is fat, so the adipocytes are almost all yellow, so a lot of lipids in adipocytes. Blue is protein, so the skeletal muscle has a very high protein content. And the red is glycogen, and that's of course highest in the liver. So the composition of these cells is different. The metabolic genes they express differ. Their metabolic functions that correlate with the tissue type function is different. Skeletal muscle being a workhorse, generating a lot of energy, whereas fat is trying to store energy. And liver trying to distribute its resources between these two and other functions. And then you can build a multi-tissue or multi-organ model like shown over here, where all of these tissue types or organ types are exchanging metabolites with the blood. And the blood is cleared and things come into the blood, like nutrients come into the blood. So you can look at a fasting and the fed state and different kinds of fed states, for instance. And one thing that came out in building this model was that you couldn't balance it without having certain buffering capabilities in the blood. So it immediately fingered some of the, the electrolyte functions in blood, and so they are explicitly accounted for in this model and are important in synchronizing metabolic function. I should say that once I gave a talk at a conference, and Mark Kirchner from Harvard was in the audience. He started the first systems biology department in the country at Harvard. And his expertise is more signaling, but he came up to me afterwards and said, and I think I'm sure he's fine with being quoted on what's going to become a public resource. But he said, listening to your talk, I was reminded of the fact that if you look at the mice, the strains of the mouse strains, the murine strains in the Jackson labs, and look at the genetic lesions in them and so forth, one of the most common, if not the most common phenotype is metabolic imbalance. And therefore, he said that his conclusion was that this system here is very hard to balance so that it behaves properly and synchronizes all the functions of organs. And of course, if you know, if you go and get a blood test, you look at the metabolite levels in the blood, and that's diagnostic for many such imbalances. So therefore, hopefully, the metabolic state in the blood that can now be characterized by metabolomic profiling will be very informative about the state of this process. And many of the wellness projects that people are now executing come down to this, is to monitor the stability of your plasma metabolome to see if you are in a steady physiological state over time. So over the coming years, maybe in your case, there will be a metabolomic profile taken annually in a fasting state, and you can compare it, and you can see if it's beginning to drift from what might be a normal state for you. Another set of markers in the blood, since we are on this topic, that is useful are tissue-specific protein. So I think we all know that in blood profiles, there is something that says liver enzymes. So if the liver is irritated by hepatitis or something, it tends to spill protein into the blood. And I believe lactate dehydrogenase from the liver is a specific isoform, and it can be detected. So like the plasma metabolome, maybe the plasma proteome will be informative about then which organ might be having problems and lysing and spilling its tissue-specific protein. So maybe in the future, the plasma profile that's done, your blood chemistry when it's done, is not going to be 2 or 3 dozen variables, but hundreds, if not thousands of variables that give you a very global assessment of what your state is. It's kind of like the modern car, when you take it for servicing, they plug in the computer, it reads all the sensors all over the car and does the diagnosis for you. So maybe something similar will happen here. A model like this is now able to simulate different states, and there's something called the Cori cycle and the glucose-alanine cycle. These are things that happen in the body that are exchanges between the liver and the muscle, and you can study them with a model like this. And then there is the fasting fed state that is shown to the left that can calculate how a liver distributes the metabolic resources that is given in the food intake. So you can now use models like this to study integrated metabolic processes in humans. There's a long way to go before a model like this is fully built and validated, but the first step has been taken. So finally, I just want to show this table here. This is a comparison of Recon 1 and Recon 2. Progress was made. I think there are about 1,800 gene products in Recon 2, and the reaction, the size of the reactome is over 5,000 now. It was 3,300, as I recall, in the first one. Well, there it is. Recon 1 says 3,700 and 7,400 reaction in Recon 2. So who knows how many more reactions will be put into Recon 3, but presumably this will build up in an iterative fashion and hopefully converge on whatever that total number of reaction is over time. So that's a summary of some of the work that has been done with metabolic reconstructions in eukaryotes. So many of them have been reconstructed and studied. They also vary in their quality and coverage, just like for the prokaryotic reconstructions. We now have, I think, a reasonably good model for yeast. Actually, that's can be stated with more certainty. There is a good model available for yeast, and there are no workable models available for photosynthetic green algae. Amongst more complicated organisms, of course, the human reconstruction is by far the best one. And it maps pretty well onto the mouse genome. You get a functional reconstruction of the mouse based on that mapping. But like I think I mentioned earlier, if you map it onto a primate, onto the chimp genome, you don't get a functional reconstruction. And that must be due to the fact that the annotation of that genome isn't good enough. I can't believe that we are metabolically more divergent from a chimp than a mouse. I think we just have better information about the mouse genome than we have about the chimp genome. So Recon 1 was published in 2007. It built on build 35 of the human genome, as I mentioned, and many applications of this are appearing. And Recon 2 appeared in early 2013, and and a number of applications of it are also appearing in the literature. So that's a brief summary of reconstruction of eukaryotic metabolism and the uses of those reconstructions. Clearly, this is going to grow a lot over the coming years, and I would anticipate in particularly the human model will be used to characterize the key disease states in human, cancer being the most obvious one. It's kind of a clonal disease and should be easier to do. Diabetes and obesity certainly I'm sure will be addressed. Cardiovascular issues will also be addressed, and people are beginning to look at that. And then oddly enough, well, surprising to me I should say, is that there is a strong metabolic basis for many, if not most, psychiatric disorders, as well as neurodegenerative diseases. And I think there'll be a lot of applications appearing there. So with that, I'll stop. Thank you for your attention.